larger structures in macromolecules. This is going to be a shorter video than my others, but very important because when we get to cells, you will need to know all of these larger structures that make up the cell components, which are all made of these macromolecules. If we start with lipids, we remember lipids are those fatty acids. They can also be the triglycerides. And in some cases, they can have a hydrophilic end, where we had, for example, a C double bond to an oxygen and then an OH. The OH is very polar. We may also possibly have a phosphate group, which has a negative charge on the outside. And remember, water loves aqueous, agua. Water loves those hydrophilic components with charges on them. Water likes things that are charged. Water will mix with things that are charged. So what happens in these particular cases is we get these charged hydrophilic heads, or in an actual cell, we have a cell membrane where we have this polar phosphate group, which has a minus charge on the outside. And then we have these hydrophobic tails, these hydrophobic areas, which do not like water. These are mostly carbons and hydrogens bonded together, non-polar bonds, and therefore, because that component is non-polar, they tend to stick to each other much more than they would stick to water because of their lack of permanent charges on the molecule. This is important when we get to cells themselves, because the cell membrane, which is one of the most important structures in preserving the cell, has a polar component, the phosphate group, and then we have a lipid component, which is nonpolar. These nonpolar regions tend to be on the inside of the cell membrane, whereas the polar part is on the outside. So when this is put in water, what we have is this part and this part hydrophilic interacting with water, while the hydrophobic part maintains a thicker cell membrane. This structure is called the because the phosphate lipid bilayer, two layers, phospholipid bilayer. On my cell, we see these in soap. How do soaps work? Soaps clean oil or grease or fat or something that's non-polar off of our skin. How does that work? Because this part of the soap mixes with water. This part mixes with the oil on your hand. Once the oil comes in contact with this micelle, the oil will be transported to the hydrophobic component. It'll be trapped in here, the dirt or the grease or whatever happens to be on the inside, and it'll be encased around the hydrophilic component, which will keep it dissolved in water. So this is how something like an oil can sit inside of the micelle, which has a hydrophobic component, and it can also remain soluble or dissolved in water because of these hydrophilic components that will allow water to carry it away. Carbohydrates, we have some structural molecules, one of them being cellulose, the other being chitin. If you notice, the starch that we looked at previously had glucose, but the glucoses were all the same repeating subunit in the exact same position. With cellulose and chitin, we have the glucose molecules flipped from one side to the other. This is called a beta linkage, whereas in the starches it was called an alpha linkage. Now you don't have to memorize that, but what you need to know is the cellulose and chitin are basically glucose molecules. Now, chitin does have one side group that is a little bit unusual, but it's basically glucose, or in cellulose, glucose. This is a molecule, a monomer, a monosaccharide that we would find, and if we could break it, we could use that for energy. We break down glucose to get energy in our bodies. However, the way these things are linked makes it very difficult for anything beyond certain types of bacteria to break these bonds. Therefore, cellulose, even though it's made of glucose just like starch, cellulose is not accessible to most living things without a bacterial a bacterial organism that lives inside of an animal that can actually break down the cellulose. We talked about cows and horses and rabbits all have the bacteria that can break down cellulose, so they're actually able to use it for nutrition. Chitin we find in the exoskeleton of insects, of spiders, of crustaceans. It's a very hard substance, also very hard to digest. Very few animals can actually digest chitin, although there are bacteria that will. Now, proteins have multiple different structures. This is where it gets quite a bit confusing. We have the primary structure, the very first one, which is just the sequence of amino acids. So we've seen that very much a very introductory way of looking at the amino acids, but we see that we have an amino acid like glycine or serine or leucine or isoleucine. These are different amino acids. So the primary, the very first structure is that order of the amino acids, which we don't necessarily see beyond that first structure, because there are other interactions that happen after the initial sequence of amino acids, whether it's glycine, leucine, valine, just in a line. From there, we'll actually have carbons and nitrogens and the central carbons all linking, interacting with each other because of some hydrogen bonding. If you remember, this is what the main core of an amino acid looks like. It has the amine on one side, it has the acid on the other side, and this hydrogen can actually interact with these oxygens, because oxygens are very electronegative, and this can actually make one of two secondary structures. So primary structure is the sequence of amino acids. From there, we get into a secondary structure. One of these secondary structures is this nice curl, which we call an alpha helix. Another one is the beta pleated sheet. So these are kind of stepwise looking sheets that certain protein molecules can form. Those are two of the possible secondary structures for a protein. Protein structure determines everything about the protein. We're made of a lot of different proteins for a reason, because they have all different structures, they do all sorts of different jobs, and that's something that we're going to be seeing many times this year. It's one of the basic foundations of biology, are all the different structures that proteins may have. From the secondary structure, we get to a tertiary structure. So you go from primary to secondary to tertiary. First, second, third. Tertiary is the third sort of structure that can happen. Now, this is something I do want to do an activity in class about this, but here is our polypeptide chain. So these are our amino acids, also called peptides, strung together, and these amino acids are strung together with peptide bonds. What does this mean? It means that the amino acids are in a certain order. All of these special groups coming off of the amino acid, remember, there are 20 amino acids. They're all made of a central carbon with a hydrogen coming off of it, a nitrogen, and another with two hydrogens coming off of it. That's the amine side. A carbon with an oxygen and an H. That's the acid side. That is every amino acid. Then there's this R group, this other group that could be polar, could be nonpolar, could contain sulfur, may not contain sulfur. That's all important for the interactions between different amino acids on the protein chain. So based on the order of the amino acids, they may form an alpha helix or a beta pleated sheet. This one looks more like an alpha helix to me. And then these side groups, these things that are sticking off of each amino acid, if there are two sulfur-containing amino acids, those sulfurs may actually form a bond between them. If there's a polar subgroup on each of these amino acids, we see this is polar because of the oxygen and the nitrogen. This is polar because of the hydrogen and oxygen. There's a hydrogen bond forming there. If there are two polar molecules, they may also stick together. Now, occasionally one of these hydrogens will pop off of an OH and hop onto a nitrogen. I believe I mentioned that in the last video. Don't worry too much about the ionic interactions right now, but know that we have polar interactions. We also have nonpolar interactions. Remember, carbons and hydrogens do not have polar bonds. So in this case, we have two nonpolar molecules, both of them hydrophobic, meaning they're scared of water. They're getting pushed to the inside of this protein molecule because they would rather interact with each other than react with a very polar molecule like water. After the tertiary structure, which we will do an activity in class with a pipe cleaner that I'm hoping will help reinforce the tertiary structure, which is the third, we get to the quaternary structure, which is the fourth. Now this kind of reviews all of the ones we've talked about. So here we have my sequence of amino acids. Each amino acid has a central carbon, has that hydrogen, has the R group, has a nitrogen. This with one hydrogen, this would be another hydrogen, except this is connected by a peptide bond. We have a carbon on the other side, can double bond it to an oxygen, and then this should be bonded to an O and an H to make the acid component. But if you remember, the OH on this side and the H from this nitrogen formed a water molecule through dehydration synthesis and ended up linking together with a peptide bond. Remember that peptide bonds link amino acids together. So from our primary structure, which is the basic linkage of one amino acid to another, we come to a secondary structure where these amino acids, in this case, they form an alpha helix because these nitrogens and these oxygens start making some hydrogen bonds with each other. And based on those interactions, they can either form this jagged beta pleated sheet or the alpha helix. The alpha helix is more common in most proteins. From there, from this alpha helix, for example, they form a three-dimensional structure. You might have your hydrophobic portions on the inside. You might have your disulfide bridges, two sulfurs linking this together, and then your hydrophilic areas might be on the outside because they like being with water. This is one protein. In a quaternary structure, you can have more than one protein sticking together. The most important of these four protein larger structures, now they don't have to be four, this just happens to be four. The four protein structure is hemoglobin. This is the protein that carries oxygen around in your blood. Each of these has what's called a heme group, which contains an iron atom that grabs onto oxygen, and this is what carries the oxygen throughout your body. We also have larger structures with nucleic acids. Now we remember nucleic acids are made of nucleotides. Nucleotides have a 1, 2, 3, 4, 5 carbon sugar. They have a nitrogen-containing base called a nitrogenous base, and they have a phosphate group where one phosphate group is linked to the sugar of another group from dehydration synthesis. We had an OH coming off of here originally. We had an H coming off of here. The H and the OH came together to form water, and then we've linked these two nucleotides together. What I haven't mentioned previously is that these nucleotides will have hydrogen bonds between them. So these are all covalent bonds. Some of them are polar covalent, some of them are non-polar covalent. But the hydrogen bonding is when you have an oxygen or a nitrogen with a partially minus charge sticking to a hydrogen bonded to a nitrogen or an oxygen on another molecule. This is why when one water molecule sticks to another water molecule, it's due to hydrogen bonding. What we see here is that two of the nucleotides in DNA, and we have 4 of them in DNA, called cytosine, which is represented by the C, guanine, which is represented by a G, thymine, which is represented by a T, and adenine, which is represented by an A. All of these have hydrogen bonds between them. This is one strand of DNA, this is another strand of DNA, and these strands stick together to keep your DNA from getting interfered with as far as any processes. Your DNA has to be very tightly regulated because this is the code, this is the copy to turn on and turn off anything that operates within you. You do not want DNA getting damaged or in some way misactivated because that's when things like cancers can form. Adenine and thymine, A and T, have 2 hydrogen bonds between them. Cytosine and guanine have 3. This means that guanine and cytosine will bond together. Adenine and thymine will also bond together. Adenine will not bond with cytosine. Thymine will not bond with guanine. Anytime cytosine is on one molecule, it'll be bonded to a guanine on the other molecule. This is for DNA. A bonds with T, C bonds with G. In RNA, which is a similar nucleic acid, although it's usually one strand, when RNA is making a copy of itself, it can actually bond with a different nucleotide, different nitrogenous base called uracil instead of thymine. What you really need to worry about, we'll talk more about RNA later on, but what you need to know is adenine makes 2 bonds with thymine, so does uracil. So these two match up quite nicely, whether it's DNA or whether it's RNA. And these are some activities we're going to do more in class. Hopefully it'll be much clearer once you do a hands-on activity. That concludes this particular presentation, and it's slightly shorter than the others. There will also be the CK12 assignment, so please move there once you're done this video. Thank you.